Hello, this is Caius Jaden. Our research project is development and application evaluation of GNSS-based fluorescence analysis kit for microplastic analysis in Arctic water environment. It is the contents. First, it is the introduction. Second, materials and methods. Third, the results. Fourth, the conclusions. And the last, our future direction. First, it is the introduction. As you can see the news, microplastics are also found in drinking water. And the accumulation of microplastics in the body adversely affects our health. But it is the current methods and limitations. We use the machine on the top right to detect microplastics, called an FTIR. It is a very expensive machine. Also, it requires specialized personnel. So, our research objective is we're going to develop a smartphone-based kit for analyzing microplastics in drinking water inside PET containers. Next, it is the materials and methods. Development of fluorescence analysis system. We're going to combine the cellophane. So we're going to make the closest peak to 614 nanometers and 550 nanometers. Next, we're going to develop an optical filter using carbon quantum dots. Second, development of quantitative analysis software and application. This is the order as you can see in the bottom. Third, kit development and performance validation. This is the kit on the top right as you can see. Fourth, field application test. We're going to choose the sea in Korea that is mostly related to the Arctic. We're going to sample it and pre-treat it. Next is the results. First, the graph. It is the smartphone light source spectroscopic characterization graph. The smartphone flashlight shows a peak near 450 nanometers. Next, development of optical filters using cellophane film, the results of that. We chose the red cellophane film only. It is because the peak is 607.6 nanometers, which is very close to 614 nanometers. Also, the FWHM is the lowest among all. We chose the neon yellow plus yellow film combination for 550 nanometers because of those reasons. Next, development of an optical filter using carbon quantum dots. This is the synthesis results. 3-minute synthesized carbon quantum dots shows the highest fluorescence intensity. Second, development and analysis of carbon quantum dots-based optical filters. The peak was 572.2 nanometers and the FWHM was 106.1 nanometers. Third, validation of excitation light filter performance. The peak was 551.4 nanometers and the FWHM was 149.8 nanometers. Next, these are our graphs of linear regressions. We chose the HLS color space H channel, which is K on the board. R square is 0.9987. That makes the most reliable predictor among all channels. Next, it is the development of analysis application. We sampled and performed image stacking to enhance signal quality and pre-processed with OpenCV and generated masks to calculate average fluorescence intensity. Mobile analysis results are transmitted with GNSS location data to a web page and integrated with seawater environmental data via API. And all data are managed in the Firebase. Next, this is our kit. All parts were fabricated from PLA and painted black to block external light. We chose a camera adapter, includes red cellophane to filter emission wavelengths. The flash assembly contains PVA CQD film and neon yellow plus yellow cellophane to emit filtered excitation light. The bottom is our kit performance validation. The kit for this study has a little more inaccurate than the old methods, the fluorescence spectrophotometer. But the coefficient of determination is much higher of 0.9952. Next is the results of our water sampling. We chose the Sokcho nearby, high transparency, which is 6 meters, and low suspended solids, 4.9 milligrams per liter. Some Jeju sites show very high suspended solids, which is higher than 10 milligrams per liter. That's why we didn't choose Jeju. Next, 20 points sampled in Gosung and Sokcho, August 15, 2025. The temperature was about 25.7 and to 29.1 degrees Celsius. The saltney was about 22.1 and 32.9 PSU. The field application test. Microplastic concentrations were measured at 20 sampling points, showing an average concentration of 0.5236 milligrams per liter. The highest concentration was at point P20. It was 1.1005 milligrams per liter. The lowest concentration was at point P8. It was 0.0821 milligrams per liter.
Next is conclusion. First, developed a GNSS-based mobile fluorescence kit for rapid and quantitative microplastic analysis in Arctic waters, combine low-cost optics and smartphone imaging to enable real-time on-site detection. Second is showed strong performance comparable to lab instruments. Third is measured average concentration 0.5236 milligram per liter across 20 sites with P20 the highest and PA the lowest. Last is verified field applicability and reliability through Arctic and ESC experiments. Last is future direction. First, improve daylight staining. Second is enhanced detection sensitivity on ultra-low concentrations. Third is establish a standardized calibration and cross-validation system. Last is introduce multi-fluorescence dyes and spectral separation algorithms. Thank you for listening our presentation. Thank you.